Here I'm going to be going over the PCR overview, which stands for Polymerase Chain Reaction. And what this basically is an efficient way to amplify sections of DNA. So again, what is PCR? Well, PCR stands for Polymerase Chain Reaction, and it's an in vitro technique used for the amplification of a region of DNA whose sequence is known or which lies between two regions of a known sequence. So it's taking not just the whole DNA necessarily, it's taking a small portion and amplifying that particular region of the DNA. And it does this many times, and it can really amplify exponentially the amount of DNA fragments that are produced. So what made this possible was one particular key discovery. This key discovery was actually occurred in Yellowstone National Park. You may think about what does that have to do with a laboratory technique used for microbiology and biochemistry? Well, it's a thermostable bacteria was found. So in the hot springs of Yellowstone National Park, they found a heat-tolerant polymerase, which prevents denaturing in the running of multiple cycles of this PCR process at high temperatures. So the PCR requires these high temperatures. Most DNA polymerases would denature and would lose their function. However, here, this bacteria in these hot springs is found to have a thermally or a heat-stable polymerase, allowing for this PCR process to be able to carry it on. This polymerase is then isolated and is known as now Taq Polymerase. So some of the required components for PCR are DNA template, primers, enzymes, dNTPs, magnesium, and some buffers. And all these are mixed in that little tiny tube to allow the PCR process to go through its natural sequence. So starting with that DNA template, well, it's DNA containing a region to be sequenced. The size of target DNA can be amplified up to 3 kilobases. So again, looking at that, not the whole DNA, we're looking at a section of that, the template of that. So we have this kind of primers that are going to match up to that DNA template. And these primers are going to be used to match up to that template are usually 20 to 30 nucleotides long, synthetically produced, and complement to the 3' ends of the target DNA. So you've got that whole sequence of DNA, we only want a small portion of it, so we use primers to kind of initiate that area where that target sequence is located. 2 sets of primers that are not complementary to each other are used because remember we have those 2 strands to our DNA. We have an upstream and a downstream primer that will be utilized. 40 to 60% GC, that's guanine and cytosine content, is preferred for better annealing because those have 3 hydrogen bonds, so they're able to hold together a little tighter. The temperature of primers can be calculated based on the nucleotides. If it's a lot of G to C bonds, it's at 3, it'll be a little higher temperature. If it's an A to T bonds, it'll have 2 hydrogen bonds, a little less temperature. And this will be outlined in a protocol. So that Taq polymerase that I mentioned, it's important. Well, this Taq polymerase, or really any one of the natural or recombined thermostable polymerases, need to be used. It's stable at temperatures up to 95 degrees Celsius, and Taq polymerase does not have proofreading, which is one downfall. But we're not looking at really replicating these DNAs to be utilized in the cell. We're looking at them to be used to be run in, for example, gel electrophoresis. Now the PCR cycle is a cycle and it does repeat. It's comprised of 3 steps. We have the denaturation step where we increase the temperature to about 95 degrees. That causes our double-stranded DNA to kind of separate. Then we have the primer hybridization where we cool the temperature down a little bit to anneal those primers, get those primers to stick on to those since separated DNA sequences. And then we have the DNA synthesis primer extension where we're kind of going through, we heat it back up to speed up the process, and we have those DNA sections extended and then replicated. And we'll go through a repeat of this process again and again. Now that first part that I mentioned, the denature of the template strand DNA by heat at 95 degrees, it's taking our double-stranded DNA, heating up so it's denatured so it's being separated. And it could be 90 to 95 degrees Celsius depending on the specific region, depending on the A to T bonds or G to C bonds presence and the percentage of those. Then we have the temperature is then lowered, and the primers are annealed to the target sequence. So if we get the temperature at 95, everything would be bouncing around so quick, those primers would not be able to anneal. Here we cool it down to 45 to 60 degrees Celsius to give a chance for those primers to stick onto the initial starting point of this target DNA sequence. The reason why there's a range of temperatures, again, it depends on how many, what percentage of G and C bonds that may be present or A and T bonds. Step 3, at 72 degrees Celsius, the Taq DNA polymerase catalyzes the primer extension as complementary nucleotides are incorporated. So we heat up to approximately 72 degrees Celsius, and this allows these nucleotides, this polymerase, to go through and attach these nucleotides to now make two new double-stranded sections of DNA over that target sequence. And this is the end of the first cycle. So the end result is 2 copies of the target DNA sequence. Seems like a lot of work to go through that one little cycle. However, once we go through the second and the third, fourth, and the fifth, this increase becomes exponential. So typically, rarely do you see a PCR run for more than 30 to 40 cycles because you have literally millions of copies of DNA at that stage. So this target amplification, as I mentioned, this is an exponential amplification in DNA. So how does that look? Well, here's our first cycle. Then each one of those are separated. So we get 4 copies, and we go to 8 to 16 to 32 to so on and so forth. At 40 cycles, if you reach that number of cycles, you can get about 1,000 billion copies from just ideally one original template strand of DNA. So this is kind of that standard thermocycle, as I said, 94 or 95 degrees to denature, only about 45 seconds at that temperature. Then we cool it down for about another 45 seconds to allow those primers to anneal. Heat it up to 72 degrees Celsius for about, again, under a minute. Let an extension phase to occur, and then repeat and repeat and repeat. At the very end, it will chill down to 4 degrees Celsius and hold there until you go through and pull your samples out of the thermocycler. As I said, this is an exponential increase, so try to give you an idea of these PCR cycles. This shows the temperature fluctuations that do occur. This kind of gives you a visual representation of what's going on, and it shows you the number of PCR cycles and the number of target copies of that DNA template that are made. You can see initially we go 2 to 4 to 8 to 16, and it dramatically will increase. And we go to 30 cycles here, we could see just exponentially how many more cycles, or based on those cycles, how many more DNA sequences we are able to get. Now, this is great when we have a lot of samples. You think, why would you want that many samples? What really makes it easy for the next step, which is gel electrophoresis. This is the sequence of using those amplified fragments, run through a gel, and then going through the southern blot process to be able to visualize those for comparisons. Now, this PCR amplification, the genome mapping and gene function is determination, biodiversity studies it can be used for, or evolutionary studies, diagnostics, parental testing, genetic diseases, early detection of cancer, viral infections, many things like that. Detection for drug resistance, forensics DNA can be used for DNA fingerprinting, so you can see that this same basic process can be used for many potential applications. The general advantages of PCR are that it's automated, it's fast, it's reliable, it's reproducible results. Once you purchase the machine, which can get expensive, it's all self-contained, and you basically can set everything up, dial everything in, and basically walk away. It's also very contained. It's used on usually a lab bench. There's less chance for contamination to occur. It's a very high output, it's sensitive, it's broad use, defined, and easy to follow protocols. If you're starting in a microbiology type lab, if you're starting in kind of a genomics lab, odds are you're going to be running some form of PCR. Because of all of these advantages, it does become very widely used and is very much an accepted and a good protocol to learn to be able to perform.